All right, hi, my name is Matti. I am doing research in the molecular and cellular biology department this summer with the Summer Research Institute, and my research focuses on the Chlamydomonas and researching their eye spot to look into cellular symmetry and how that helps the cell move. So just a little bit of review for you guys. The Chlamydomonas are the green algae. They're unicellular, so each organism is only one cell, and we focus on the eye spot, which is right here. There are other organelles inside the cell that are of importance. Mostly these microtubules, the four that extend down, are like a skeleton for the cell, so they help it with its structure stay in the same spot every time as far as where things develop. And also the basal bodies, which are like the organizing centers for the cell, they communicate things down the different microtubules and communicate to the flagella to move. The two flagella extend out from the cell, work like wings to kind of help the cell move along, and they all get their information about light and movement from the eye spot. And the eye spot is composed of granules, which come from the chloroplasts that kind of orient the cell on where the light's coming from, and photoreceptors, which are along the plasma membrane, and those work to kind of bring the light in, and it sends up an influx over to the basal bodies to say, hey, there's light, we need to move this direction. And the reason they the reason they move for the light is so the chloroplasts, which are all at the bottom of the cell in here, the cell moves so that the chloroplasts face the light, because that works for photosynthesis, and that's how the cell gets its energy. So studying the eye spot can help learn about the asymmetry of the cell and how it moves, and that benefits scientific and medical research. So what I did in my research is I worked with a double photoreceptor mutant. So the two photoreceptors which are along the plasma membrane are CHR1 and CHR2. So I worked with a mutant that was coded to not have those photoreceptors. And the purpose of this is to find out, how does the cell function without its photoreceptors? Does the eye spot still work? And can the cell still move? So to begin this process, the method I used was reverse genetics, which is where you create a mutation in a gene to see what happens using the known gene. So you have a gene that you know about, and you want to get rid of it to see what is its role in the entire cell. So I start by generating thousands of strains of mutants, trying to insert a gene into another gene. So right here, this kind of describes it well, is I start with this ARG gene, and I want to target the gene for the photoreceptors. So I put a gene into there which disrupts this gene, and I can't necessarily just take the one gene, put another one, and my problem be solved. It doesn't work that easy. So you just have thousands of strains of these mutants trying to get the ARG gene to insert into my gene over here. So then there are, like I said, multiple different things that could happen. It could not insert at all. It could insert in a completely different location. So that's why I test them for phototaxis, because if my gene inserts successfully, then that means that my cell won't phototax. And basically, what is phototaxing? It's the response to light. So I talked about at the beginning how the channel in the photoreceptors communicates to the basal bodies to move. So the light comes in, and the eye spot tells the cell where the light's coming from, and they want to move so that the most amount of light is towards the bottom of the cell where the chloroplasts are. And so I do a phototaxis test to see if they move or if they're not moving, to let me know if I got my mutant. So how do I start? I start with a Chlamydomonas that has a mutation in the COP3 gene and the ARG7-2, which the COP3 gene is for the CHR1 photoreceptors, and that's a mutant that scientists have already constructed. And the ARG7-2 is a media that we use for the cells to grow on. And this means that I can't grow these cells on a plate without supplementing them with ARG. So if I take the ARG away, none of them will grow. If I put the ARG back in there, only the ones with the mutation will grow on the plate. So I use a vector which carries the ARG gene into the insert, into the COP4 gene, and the COP4 is for the CHR2. So ultimately, if my ARG is successful, only the things that have the mutation will grow on the plate, and then from there, I take the ones that have the mutation to not phototax. So I'm looking for two different characteristics in my final product. So like I said, they won't be able to phototax, and what I do after I've hopefully inserted my gene is I get the culture that's on the plate, and like I said, there's like little dots of cells that are the ones that successfully grew on the plate with the media. So I pick those, transfer them into a minimal media, and then let them grow up for a few days, and then transfer them into a different media to watch if they can phototax. And if they don't phototax, that's good news, but I like to double check it. So we concentrate them, regrow them, and then rescreen. And if we find any that don't phototax on the second screening, we look at them under the microscope to look for eye spots, if they're regular, irregular, see what the cells look like, and then also make their genomic DNA to do a PCR reaction. So making the genomic DNA, I start with a culture of the cell that I think is mutated, just to get as much of it as I can. I spin the cell down in the centrifuge and add a death buffer, which eliminates all the cell and gets me most of like the DNA product that I want. I incubate it overnight, add ammonium acetate, phenol, and chloroform, and spin down, add chloroform to extract everything but the DNA. And then I wash it and resuspend it with water, and that makes it ready for the PCR, which is a polymerase chain reaction. So this basically amplifies the targeted sequences that I want with my DNA. And there's 3 steps to it. It all happens in a machine, but basically it denatures the DNA to make it single-stranded, attaches on primers that my lab has selected for me to use, and then extends the strands and repeats it for 30 cycles. So that way I get to see a DNA product. And just a little bit of preview results for you guys to kind of connect in with the methodology. I did find a mutant in my screening. So if you look at the wild type picture, there's kind of an orange on the side, and that's what the eye spot is. It's made of carotenoid granules, so they come up orange under the microscope. And if you look at this one, our microscope doesn't do color, but there's nothing along the sides that would show an eye spot. So that means that my mutant doesn't have an eye spot. And when I did PCR, it did give me, oh, I'm pointing at the computer. It gave me a product, which means that the 2 primers I use they're close enough. They formed a product when they're amplified, which is very good news for me. So those are my acknowledgements. Thank you, everyone. And does anyone have any questions? Get this thing off of me. Okay, whenever, yeah. Do we have any questions?